Welcome to Wales Tech Week 2021. Thank you to our partners. Enjoy the session.
Prynawn da, croeso I arddangos Prifysgol Aberystwyth. Warm welcome to the Aberystwyth University Showcase. My name is Helen Jones, and as the Director of Research, Business and Innovation, and essentially the university's gateway for collaborating businesses, it's a pleasure to be speaking here today. I don't have any slides. I thought you might all welcome a break from PowerPoint while I give a little background on the breadth and quality of research at Aberystwyth, and also introduce you to some of the academic experts at Aberystwyth University who talk about how their research and technology partnerships with businesses are realising some great industrial advances and societal benefits for Wales and beyond. Aberystwyth is a research-led university, and though it wins accolades for its teaching, so in 2020 it was the Times Good University Guide Welsh University of the Year, and then this last year in 2021 it was the Times Best UK University for Teaching and Student Experience. It's the research that ensures the quality of teaching, and that is also recognised as excellent. So in the last national assessment for research excellence, over 95% of its research was of an internationally recognised standards for activity, outputs and its impact. It does this over a broad range of disciplines in natural, physical, data and social sciences, and it also has a strong presence in the arts, creative and cultural themes. As a research institution, its strengths are in its connection with its collaborators and its communities, its cultural integration and its climate consciousness, and it does this all bilingually in Welsh and English. The rural and coastal location of the university, as well as its long-standing connection with the land, environment and local economies, has developed a great research capability in agriculture, health, communication, human, political and landscape geography, as well as the underlying technologies that advance our knowledge and application of our knowledge in these areas. Agriculture is a good example. We host the world-class Institute of Biology, Environmental and Rural Sciences, and we mirror its research capability in our teaching and innovation facilities. For example, we're about to launch the only vet school in Wales later this year. And as part of our Vet Hub Innovation Cluster, we host containment level 3 laboratories and testing facilities, which are pretty rare. We have the AberInnovation Centre. It's designed to work with and connect with businesses with our agri-food research. There's a Wednesday session coming from AberInnovation, and I'd encourage you to go to that. I think it's at 2 o'clock in the afternoon. Our research in wireless communication, spectrum and sensing technologies also play a big part in our innovations for low carbon, green energy and climate change solutions. So the Department of Research, Business and Innovation, which I direct, facilitates the business interactions with our research on many levels and in a variety of ways. We support researchers in their strategic self-development, which includes capturing external funding for research and exchanging knowledge with industry as well. We facilitate partnership development, as well as manage the operational and governance elements of research, such as costing, accounting, ethics and contract management, project quality and compliance, reporting and commercialization. We also support fledgling businesses as well, whether they're from our students, our graduates, our staff, whether that's startups or spin-outs. If you're a business and you're keen to work with us, search for us online quite easily. You just type in Aberystwyth RBI into the Google search bar, and we'd love to hear from you. We can direct you to wherever you need to go within the university. Now, for 2 great examples of Aberystwyth research and collaboration, the first is from David Bryant, who will share with us his international project focused on turning industrial food waste into valuable products of the future, some of which you may have already heard of. And secondly, Otar Akanyeti, who will share with us his research on how technology can play a positive role in stroke recovery. There's about 10 minutes for each, with questions as well too. So David, if you're ready, please continue. I think you're on mute, Brian.
I am, yeah. Thanks very much, Helen. I'd just like to share my slides. So I lead a project called BioReview, which is biorefining value from industrial waste. It's funded by the BBSRC, Newton Bubba project, DBT in India, and Innovate UK. And it's an academic and industrial consortium looking to valorise components of waste out of the sugarcane industry. And in the UK, the industrial partners are led by Manrochem, who are a chemical engineering company who build and design and build factories. Membranology, who are a Welsh downstream processing company who have expertise in cross-flow filtration and reverse osmosis. Free Energy, who are a Welsh anaerobic digestion company. They build, install and operate AD units for farms, etc. In India, our industrial partner is Gayatri Sugars. They have a couple of sugarcane mills. The one that we're working with is based just outside of Hyderabad in India, and they crush somewhere around about 6,500 tons of cane a day. To make sure that what we're doing has some environmental benefit and we're not just making things worse for the environment. We have LCA Works, who are doing greenhouse gas emission studies, techno-economic analysis, and also the socio-economic impact. So why is our Industrial Waste Challenge project focused at the sugarcane industry? Well, India is the second largest producer of sugar in the world, Brazil being first, and they also have a very extensive bioethanol industry from sugarcane, where they ferment the molasses through to ethanol. So there's two main waste streams here. One is the sugarcane bagasse, which you can see here, this fibrous residue, and currently that's burnt to generate heat and power. But associated with that energy production are particle emissions. So this shows you a map of India, and these particulate emissions are all associated with sugarcane refineries, which obviously have an impact on the respiratory health of the local population. Now, in terms of ethanol production, they produce, well, the sugar mills in India in 2016 produced somewhere around about 1.75 million cubic meters of ethanol. But there's a huge cost to that. There's a generation of what is 14 million cubic meters of spent wash waste, which is basically wastewater. To put that in some kind of perspective, that's somewhere over 5,500 Olympic swimming pool worth of wastewater every year. And if that is indiscriminately goes into the water courses, etc., it causes a huge environmental burden. So these two areas are where we're primarily focused on trying to produce a bioproduct portfolio to try and see if the companies can invest in making money from waste streams. So the four main products that we're looking at are Indian partners at the Indian Institute of Chemical Technology, led by Venkata Mohan, probably India's leading waste biorefining expert, is microcrystalline cellulose. And you can see there that's got a market projection of about 1.1 billion by 2025. With regard to the spent wash, we're looking to produce volatile fatty acids. Again, that's in association with the Indian Institute of Chemical Technology, but also membranology with their commercial expertise in downstream processing, and also Free Energy with their commercial expertise in AD production and when to stop to produce the volatile fatty acids. We're also looking with Bangor University to produce some cellulose depth filters for cleaning up wastewater. But primarily at Aberystwyth, we're interested in producing xylitol, which is a sweetener that's tooth friendly for kids. So the main areas that we're looking at are process integration, pilot scale biorefining, and demonstration to try and help inform business decisions. As I mentioned earlier, we're looking at life cycle assessment and techno-economic analysis, but also looking to see if we can actually improve some the social aspects of farmers and women and children in the rural area by providing upskilled employment opportunities. And by the end of the project, all this we should be wrapping up inside a commercial realization and route to market plan. This is a quick slide just showing you some of the work that they're doing in India. This has already been done. They're now on the way to fabricating a much larger reactor. But this is the team in India and some of the pilot scale VFA reactors that they've been producing. And this is Dr. Venkatamohan here. In the UK, we've got, as I said, Free Energy based up in Wrexham, and also Membranology. So here we've got Chris Morris, who's director of Free Energy, and we've got Dr. Bob Lovett, unfortunately passed away last year, who's director of Membranology. And this is just, well, this is the downstream processing unit that they've built up at Free Energy that is processing their anaerobic digestate through to a partially purified VFA stream and the digestate that's left behind. So as I mentioned, we're mainly interested in the xylitol bioprocess that we've developed at Aberystwyth. So we're engaging in strain optimization and process scale up. So we basically have been trying to improve the productivity rate so that we can use sugarcane bagasse hydrolysate, and I'll explain a little bit about that in a minute, to produce xylitol. And we've been using a combination of bioprocessing improvements and also some synthetic biology approaches to engineer Candida tropicalis to improve the titer so we can get above 10% weight for volume in the fermentation. And then we'll be scaling up this process to around about 250 litres. So the main thing that we're trying to do is improve the titer and the rate. Now the yield, we've already engineered the organism. So for every gram of xylose that goes into the organism, 1 gram of xylitol will come out. So a little bit of background about what xylitol is. It's a sugar alcohol. So it's a natural sweetener that you find in fruit and vegetables, but it's at vanishingly low concentrations. But it has very good applications in dental, dietary and diabetic applications, because in dentistry, it inhibits the causal agent of tooth decay in children. So it inhibits an organism called Streptococcus mutans that's responsible for dental caries and rotting that you get in kids' teeth. In dietary terms, it's only got 70% of the calories of sugar. And for diabetes, its metabolism isn't insulin dependent, so it's compatible with diabetic products. The chemical process, though, is via, or the current production process is a chemical-based process where they use hydrogenation, high acid, high temperature. It's very chemical intensive, and it's led to a whole load of environmental concerns in China, with them closing down a lot of production. So there are fermentative routes that have been produced, but so far they haven't been commercialized. So we have a strain of yeast which we call Y4X2, and this yeast can take xylose from sugarcane bagasse once we release it and convert it into xylitol very simply via an enzyme called xylose reductase. And this then can go into a whole range of health conscious products that are currently available, Holland and Barrett's or general retail outlets. But to release the xylose from sugarcane bagasse, which is very fibrous, we have to do pre-treatment. And the pre-treatment we use is something called steam explosion. And effectively, it's if you've ever done any pressure cooking, this is what this is like. It's a big pressure cooker, and we put the biomass in here. We heat it up to a high pressure and high temperature for a set amount of time, open a release valve rapidly, and that rapid decompression blows the biomass to pieces. And we've tried this on various different feedstock, willow, wheat straw, miscanthus, and also corn stover. And then we can squeeze out this juice, which then contains the sugar, xylose, which we then ferment at our fermentation facility. Now, all these pilot scale equipment are available at the AberInnovation Centre for Biorefining, and that's where we've been doing most of the work. This graph just showed you that we can optimize and tune the way that the sugar comes out. So for xylose released, the blue bar represents the monomer and the orange bar represents oligomers. So here 2 to 8 means you've got a minimum of 2 xylose stuck together, or maximum of 8. So just by altering the catalyst and the conditions, we can tune for sugarcane bagasse and or even miscanthus the type of composition that we want to achieve. And also, more importantly, is that the conditions will relate to how much other contaminants might get produced. So here we've got the main contaminant that gets produced is another sugar called arabinose. So we can adjust it so we've got more xylose to arabinose. So you can see here we've got 100% of the xylose released, but we've got an awful lot of arabinose present. Whereas here we've only got 83% of the xylose released, but most of it's present as xylose and not arabinose. And we've been working with our colleagues up in Bangor at the Biocomposite Centre, and they are scaling up that process that we've developed into a continuous process using their equipment. So this is the continuous steam refining installation up at Bangor. And just to show that we're getting similar type of yield, here we've got the DP2 to 6, mainly a mixture of oligomer and monomer in this one. But again, similar sort of release as to what we were getting before. So over the project that we've been running, we initially started off down here with a xylitol rate of just under 1.5 grams per litre per hour, and our target was 2.7. So we've been doing various different trials to take it through. So this blue bar here is the xylitol titer, and we wanted to aim up here. And the grey blobs are the price of the media. So what we're trying to do is increase productivity and reduce the amount we're going to have to pay for our media. So it's taken some optimization. And every time we make an advance and then change the media to try and get a cheaper one, we get a hit back in productivity and titer. But eventually, as we've progressed, we're now hitting over our target, which is we're now hitting about 110 grams per litre, and at a productivity which is double the rate that we'd initially set ourselves, so we're about 5 grams per litre per hour. And the media is roughly half the price of the expensive one, or we've probably shaved off, I don't know, about 30% of it from when we started. So all these data and all these improvements are based around trying to improve the techno-economic performance of the process, so thereby trying to make it cheaper in the long run to produce the product. And this just shows the area where we had to address an issue of the hydrolysate toxicity. So that's the sugars that have been released when we steam explode it. There's also some inhibitors for fermentation that are released, and we have to minimize those. Here we've looked at the promoters. So we're looking at gene expression that's going on during the fermentation process. So here's a fermentation process. This shows xylose going down, and this line shows xylitol going up. And we've taken samples at each of these time points throughout the process to look at the genes that are expressed, so that we can understand more about the organism and the process. And this just gives you an overview of there's somewhere around about 4,000 genes that are upregulated and downregulated at any one of these time points. So it's an incredibly complicated data set. But using the latest genome technologies and bioinformatics that are available, we're able to see at a glance where genes are upregulated and downregulated. So if we look at this table, this chart here, the triangles that are in blue and pointed down, that means those genes have been downregulated or switched off. And the ones where they're in red or very dark red shows that they've been upregulated. So we can then compare this with the graph that we've got to see what's going on. This is at a cellular level. So this is at a very big level in various different compartments within the cell and different locations. Well, we can look at biological processes. And if we look here, we can see there's carbohydrate metabolic processes going on that are being upregulated and some that are being downregulated, but also ones that are being upregulated and downregulated because they're quite complex sets of genes, and not all genes will be necessary for a particular function. And similarly here, we have transmembrane transport, and then we can look a little bit further to look at their molecular function. And considering we want to produce xylose by the activity of xylose reductase, we can see that gene has been upregulated in this sample, along with a whole range of other genes. So really, we're now starting to use this information to understand more about what's going on the genetic regulation of the organism to try and improve the process. And just as a final mention, we've got a startup called Architect Bio Limited, and they're now commercializing the biotech process for xylitol production that's been developed at Aberystwyth University. So they've got a few employees now, and they're based at the AberInnovation campus. So it's quite a good and exciting time. So thank you very much for your attention. Do you have any questions?
Dr. Bryant, I thought we could take questions in the comments box.
Right.
So yeah, and I'm happy to field those as they come through. And we can do them all at the end if you like. I don't know if Dr. Akanyeti, you'd like to do your presentation, and I can scan them all together. We can do questions and answers at the end. Does that sound all right?
Yeah, that sounds good to me.
Great.
Yeah,
Okay.
Yeah, I'm happy to share my slides. Yeah, so hello. Basically, in our research group, we are interested in bringing stroke care into the community to help people right there, right then, and reduce some of the workloads from the shoulders of the NHS. So we are relatively a new research group at Aberystwyth University. Our group is called Aberystwyth Stroke Research Group. We have founded the group more or less 2 years ago, and we are proud of being very interdisciplinary, and we draw from the fields of computer science, clinical exercise science, neuroscience, biology, and psychology to provide an integrated overall solutions for stroke care. And in this talk, I'm going to focus more from the computer science perspective, especially given the theme of the organizers' Wales Tech Week. And what we just to describe what stroke, it affects almost like 50 million people in the world and 100,000 British citizens every year. So there are two different types, and what happens in one case is basically there's a blockage in the brain vessels that carries oxygen to the neurons, and when the oxygen supply is blocked or stopped, neurons starts dying, and that obviously have a significant impact in the brain injury, and that leads to cognitive and motor impairments. And similarly, you can have a rupture in one of those vessels, and then there's a bleeding in the brain, and that also causes pressure in the neurons and then causes cell death as well. And that's a different type of stroke, but the outcomes are similar. And again, in people who have stroke, leads to severe cognitive and motor impairments. So just before I start describing the technology that we are trying to develop in AberStroke, just it's important to understand the timeline about how stroke care is delivered at the moment. And what you typically see is when stroke happens, obviously the first few hours is extremely important to resolve the blockage or stop the bleeding, so that people who just had stroke can release from the hospital as quick as possible with very little impairments. But if that doesn't happen and the injury sustains, then the first 24 hours is extremely critical to stabilize the patients. And during the first week, patients typically stays in intensive care. And once they're getting into more stable conditions, they're transferred into subacute wards where their rehabilitation process, depending on the type of impairments they have, starts. And these impairments can vary from language impairments or cognitive impairments or gait and balance disorders. And depending on the severity of the impairment, the length of the hospital stay can vary from 3 weeks to 6 months. And once patients are ready to or reach to a certain stage where they can be discharged from the hospital, they're ready to go home. And what happens in our current healthcare system, we see this invisible handshake where patients slowly transition from hospital or inpatient clinic care to more of a community care practices. But unfortunately today, there are significant challenges in the community care. So what we see is people usually fall off the radar very quickly. So patients are expected to come back to the hospital for follow-up controls, but the majority of them do not show up. And this may be due to maybe they feel burdened, there's nobody to take them to the hospital, or it might be just that the resources are not there, or they just don't want to do it for many different reasons. And our NHS services at the moment do not have the resources or capacity basically to follow up on these patients. And typically when patients go invisible, usually taking care of them falls on the shoulder of their family member, and this is a huge problem at the moment in the UK, and it's known as a caregiver burden. This is basically a voluntary work, but it has a huge physical, emotional, and economical consequences on the people who are providing that care, and also putting them in risk to develop other healthcare issues or health issues in the near future. And obviously, if stroke happens and there's a long-term disability, recovery from that hinges on extensive rehabilitation, intensive rehabilitation, and persistence. And to be able to provide that rehabilitation programs, patients need to attend rehabilitation programs, and these are it's almost like a life-changing event, and it has to be done in a very personal manner. And obviously that's very difficult if there are no resources. There are not also these rehabilitation programs available for patients, and that puts them also in a disadvantaged position where they may not recover some of the functionalities that they should have if necessary care could have been provided. And one major issue in this group of people is falls. And when you're disabled and when you cannot walk around very well, and when you spend a lot of time sitting, then obviously you start losing a lot of muscle mass, and then fear of falling starts kicking in. And usually we see quite a lot of cases where falls happen and secondary injuries, and then those people who had suffered from stroke at the beginning just readmit to the hospital for secondary health issues. So in our group, we see this stroke causes disability, disability causes more of a sedentary and depressed lifestyle, and obviously that will cause you more health issues, and that health issues is going to trigger more reinforces the disability, and then we see a very negative, vicious cycle of stroke that actually pushes people in the negative direction. So in our group, we are trying to bring technology and novel interventions in to break this vicious cycle. So we have some principles, and what we're trying to do is we're trying to obviously improve quality of life, health, and integration between healthcare and community care services. But also we would like to provide training and rehabilitation programs to patients in the community, and do this as remotely and as automatic as possible. And while we are doing so, we would like to be safe. We would like these services to be available to anyone, anytime, and anywhere. And also we would like them to be more personalized, so that they can be tailored to the specific needs of individuals. And this is more of the innovation side of the things, but we are also very interested in the science parts of the project, and we would like to understand the mechanisms that underlying stroke injuries, as well as motor learning skills that actually allows how rehabilitation programs help patients to rewire their neurons in the brain, and it just helps them to recover. And the more we can understand these processes, the better training programs we can design, and then we can help stroke patients to reintegrate with the society. So today, in few words, I would like to tell you a project that we are currently working on, and that's basically has two legs. And the first one is we would like to use smartphone to assess gait and balance impairments in stroke patients, and feed this information to clinicians for diagnostic as well as treatment decision making. And once those decisions are made, we would like to use the same interface, supported and powered by an artificial intelligence, to design personalized training programs and deliver those training programs through smartphones, so that we can help people who suffer from stroke to regain fitness, cognitive and motor abilities. And just to give you a little bit of a scope of how things are done currently in medical settings, one of the ways that actually clinicians assess stroke patients is using observer-rated clinical measures, and this is one example that we see quite common is used in hospital settings. It's called modified ranking scale, and as you can see, even though the patients get a score, these scores are very qualitative, in the sense that you basically assess what patients can do and cannot do in a very loose terms. But obviously, this is not the only way that clinicians use. They also have the expert opinion, and this is just an example of one of those, again, qualitative but very detailed clinical diagnostic, where we see a very medical way of describing the gait and the issues that we see in a stroke patient. And again, here what we see is it's very detailed, it's very descriptive, it's very useful, informative, but it's not quantitative. So we are missing that quantitative element about how severe the walking is, how impaired it is, what's my walking velocity, do I have an asymmetry, if so, what that asymmetry is, and how can I help it to reduce it. And obviously, there are also laboratory settings or laboratory systems that allows you to do high resolution recordings to extract motions from different ankles and joints. And usually, these 3D motion analysis systems or capture systems are expensive, and they are basically they need technical expertise to operate, and usually their capture volume is very limited. So what you tend to see is stroke participants are invited to lab environments maybe once in their lifetimes. Their gaits are measured, and that would give them more of a snapshot of what people may have been experiencing. But there's a growing evidence that how people walk in natural conditions, in real world conditions, are very different than from these lab measurements. And we wanted to basically bring some of these quantitative skills into the community with a much more accessible system that can provide us information or real-time feedback about gait impairments. And that's what our smartphone technology is aiming for. So what our technology can do, it can tell us where patients are, how often they are active and where they are going, but also we can tell how well they walk. And for those, we are extracting some temporal parameters, like how many steps per minute they're taking, what's their stride time, do they have variability from step to step, is there a gait asymmetry from their affected and unaffected side, and how much time they spend on the ground, how much time they spend on taking the steps. And here I just give you a very, very simple example between age-matched control subject, a healthy person, versus a chronic stroke patient who just had a stroke a few years ago. And as you can see that there are very, very significant differences between the two subjects in terms of stroke participant. Even though he's asked to walk as fast as possible, is able to take very few steps compared to the healthy. And there's a higher gait asymmetry as well as step variability. So what our technology can do is can just basically fall into this happy medium where it will provide some quantitative and useful measures that actually can be used in the community settings. And how do we do that? Basically, we are taking advantage of the motion sensors found in mobile phones, such as accelerometer and gyroscope. And typically, when you rotate your phone, the phone automatically adjusts its screen to help you when you're having a video conference with someone else. But we can also use the same technology to monitor the steps that people are taking when the phone is in their pocket or on their lower back. And from this, we can easily tell actually whether those steps are symmetric or how much time it takes a person to take 2 steps in a row. And what you see in this graph is just an example over a 5-second window. We are showing age-matched control subject on the gray versus black line indicates a similarity from a stroke participant. And as you can see that there's a significant variation in the peaks between affected and unaffected side, and that gives you quite a bit of an asymmetry, the way that stroke participant is walking. And as well as if you look at the time difference between the peaks, you see a longer time period compared to the gray. And that also shows you that actually they're taking slower and slower steps and maybe shorter steps too. So this is the general logic and reasoning behind our technology. So where we are so far, we have tested the system on more than 120 participants so far, and these people come from very different demographics, very young, up from 18 years old up to 80, community dwelling older adults. They also have different walking abilities, and most of them very healthy, but also we had frail or participants who were suffered from stroke and Parkinson's disease. And when we compared our measurements to gold standard method, we see a very similar result. So we are very, very encouraged. However, we haven't tested our system in outdoor and more natural environments yet. So far the experiments have been done in controlled laboratory settings, and the next step is basically to take this to outdoor environments. But also, we are just about to start an NHS funded study to test our system's capabilities in clinical applications. So we are basically inviting stroke participants to a 3 months longitudinal exercise therapy. And what we're going to do is we're going to train them 3 times a week, and we're going to look at their gait impairments at the beginning and monitor how those gait impairments change over time using this wearable technology. So this is a project in collaboration with Havel, the Health Board, and Dr. Federico Villagra and I are leading the project as PIs. And obviously, maybe not today, but just to give you a teaser, once we kind of establish the technique of assessing the gait, the next step is to use the mobile platform also as a personal trainer, and just to provide the rehabilitation programs in the community, in patients' homes, so that everybody can access it. And that's all from me, and I'm more than happy to answer your questions.
That's great. Thank you, Dr. Akanyeti. I'm just going to post in the comments section our web address is there, so people can see your projects online if they want to know some more about it, and also get in touch with you. But I'm looking at the comments section now to see if there's any questions come in, and I can't see anybody with some new questions. However, I did have a question for both of you. I appreciate we have to finish bang on Q2, but both Dr. Bryant and Dr. Akanyeti, my one question was, how did the project come about with these collaborators? Had you already worked with these collaborators or this technology in another project, or was it a serendipitous meeting of people and of minds?
So sorry, with our one, that came about through. We did have a project first, which was European funded, where we did a lot of the preliminary work in developing the organism. And then we also had, funded by the BBSRC, a travel award, and part of that funding partnership was Gayatri Sugars in India. So we've been kind of like developing relationships, and through BEACO, which is another biorefining program at IBERS, we have a relationship with Free Energy up in Wrexham. My colleague Srinivas Ravella was very instrumental in developing that relationship. He has a lot of expertise in AD and understanding microbial communities within AD reactors, etc. So it's kind of like really building upon existing relationships and developing more and more connections, really.
Great. And that would have taken years, months?
Years, yeah. So
yeah, just to give people an idea, I think, of how long these things take to foster relationships.
Yeah, for where we are, from starting with xylitol was in 2014. I think the initial grant, and now we're coming 2021. The project's coming to an end. We'll finish by the end of the year. But in that process, we've managed to spin out a company as well. So so far, we're quite pleased how we're getting on.
Yeah, sounds great. And Dr. Akanyeti, how did your project come around?
For us, it was a very different road. It was a serendipity, probably. With Dr. Federico Villaga, we were swimming in the morning, and he comes from a stroke background and exercise physiology. And I have lots of experience in AI, but never had the opportunity to apply to stroke before. But it made a lot of sense, especially for our communities here, given how little resources we have, especially in the rural Wales. And as a nice collaboration, we started 2 years ago, but when you work in the health field, things move very quickly, and there's so much interest and so much momentum. So actually, it's been growing in a very fast rate. And so,
yeah, you can respond to the demand.
Yeah.
You can answer the questions, and you can develop a device. It can move really quickly, can't it?
Yeah, and also, like when we started, we were 2, but now we have we are leading a group that has more than 10 researchers in it, PhD students, postdocs, and because not just there's a demand out there, but also a lot of people interested in working in this domain as well.
Yeah, and on your projects, have you met researchers, business partners that you thought, actually, I want to do a different project with this group, or I think we could actually go off on a tangent and develop a different kind of research innovation relationship?
Yeah, absolutely. There's, it's one of those things where you're collaborating with people. It's a fertile ground for ideas, especially as you approach things differently. So yeah, there's definitely more things that can be done.
In our case, we have started collaborating with Starfish Labs from AberInnovation company, and they are interested in looking into cerebral palsy, which is considered as a stroke in children. And there's a lot of obviously this gait assessment technology that we develop for stroke patients can be easily used for any gait impairments. So there's lots of opportunity to take it to different domains. And yeah.
Sounds great. Well, thank you both. It's been really interesting and great to hear that there's more to come, not just on these projects, but on other separate projects and relationships as well. It's great to hear. And I hope everybody listening's got a good idea of what Aberystwyth is capable of and how keen we are to work with businesses and the collaborators internally, locally and regionally. So thank you both. It's been a pleasure.
Brilliant.
Thank you for reminding us, yes.
Thank you for watching. Rewatch all of our sessions online. Thank you to our partners.